Today, we're going to follow a single, tiny piece of tissue on this incredible journey through the lab. This is the hidden, super intricate process that turns a small sample from a patient into a definitive diagnosis. So let's pull back the curtain and see exactly what happens. You know, it's a feeling a lot of us have had. A sample's taken, it disappears behind a lab door, and then a few days later, a report just appears. But that journey it takes in between, well, that's a fascinating story of biology, chemistry, and some seriously meticulous craftsmanship. And that's really the core of it all. See, the cells in that tissue have a story to tell, a story about what's really happening inside the body. Histopathology, that's the science of taking that story, which is totally invisible to the naked eye, and translating it into a language a pathologist can finally read. So how do we get that story out? We're going to follow this clear roadmap. A series of pretty incredible transformations. We'll start with how the tissue is preserved, then how it's processed into a solid wax block, sliced impossibly thin, and finally stained to bring all its secrets to light. All right, our journey kicks off with step 1, preservation. And you have to understand, as soon as a biopsy is taken, it is a race against the clock. That tissue is no longer part of a living system, so the countdown to decomposition starts immediately. See, the very moment a tissue is removed from the body, its cells begin to die and break down. That means all those delicate structures a pathologist absolutely needs to examine could be lost forever. So the first, most critical mission is to basically freeze that biological moment in time, preserving the cells exactly as they were. And this process has a name. It's called fixation. It's basically a chemical bath that slams the brakes on all biological activity. It stops enzymes from chewing up the tissue, it kills off any pesky microorganisms, and it hardens the sample, which is going to be super important for the later steps. So how does this fixation thing actually work? Well, chemically speaking, there are 2 main ways to do it. The most common method uses what are called cross-linking fixatives, like formalin. You can kind of think of it as building a tiny scaffold inside the cells. It creates these chemical bridges between proteins that just lock everything in place. The other way uses dehydrating agents, like alcohol, which, just like it sounds, pulls all the water out, causing the proteins to harden. But here's the thing. This process isn't instant. Formalin, which is the real workhorse of the lab, seeps into tissue incredibly slowly. We're talking at a rate of about 1 millimeter per hour. That's why a sample might need to sit for a whole day just to make sure every single cell is perfectly preserved. Okay, so our tissue is preserved, but it's still soft, and it's still full of water. And there's no way you can slice something like that thin enough to see through it. So this next phase is all about a pretty radical transformation, turning that soft tissue into something solid enough that it can be sliced with microscopic precision. This transformation happens in 3 key steps. First, we have to get all the water out. That's dehydration. Then we have to clear out the chemical we just used for dehydration. And finally, we infiltrate the tissue with our support medium, which is paraffin wax. So, why do we need to remove the water? Because our end goal is to get wax into the tissue. And as we all know, water and wax, they do not mix. So the solution is dehydration. The tissue gets a bath in a series of alcohol solutions, starting weaker, around 70%, and then gradually going up to 100%. This slow and steady process gently pulls the water out without shocking and shrinking the delicate cells. Okay, so we solved one problem, but now we've created another one. The tissue is full of alcohol, which also doesn't mix with wax. We need a middleman, a go-between. This step is called clearing, and it uses a chemical, usually xylene, that can mix with both. And here's a really cool side effect. As the xylene replaces the alcohol, the tissue becomes completely transparent. All right, now for the grand finale of this processing stage. The now transparent tissue gets placed in a bath of warm, molten paraffin wax. The wax slowly seeps into every single nook and cranny, filling every cell where water and alcohol used to be. Once it's fully infiltrated, it's placed in a small mold, more wax is poured around it, and it's left to cool. And what you get is a solid block of paraffin wax with the tissue perfectly suspended inside, kind of like fruit in a Jell-O mold. So our tissue is now ready to be sliced. But even after we cut it, the cells are still completely colorless, totally invisible. This next stage is where the science really becomes an art form, as we get ready to paint the tissue with special dyes to reveal its hidden architecture. This is done with an amazing machine called a microtome, which shaves off these incredibly thin ribbons of wax. We're talking just a few micrometers thick, way thinner than a human hair. These delicate slices are then floated on water and picked up on a glass slide. But if we looked at this under a microscope right now, we'd see a ghost. Almost nothing is visible. So how do you make the invisible visible? You stain it. And the undisputed star of the pathology world, the absolute legend, is the hematoxylin and eosin stain, or H&E for short. For over 100 years, this two-dye combo has been the #1 tool for visualizing tissue structure. The power of H&E is that it's a tale of two dyes with completely opposite properties. Hematoxylin is what we call a basic dye, so it's attracted to the acidic parts of the cell, mainly the DNA inside the nucleus, and it stains them a deep blue or purple. Eosin is the exact opposite. It's an acidic dye. It's drawn to the basic stuff, like the proteins in the cytoplasm, and it stains them in all these beautiful shades of pink and red. Okay, we've preserved it, processed it, sliced it, and stained it. After this long and incredibly careful journey, our small piece of tissue is finally ready for its ultimate purpose, to tell its story to the pathologist. It's amazing, isn't it? What started as just a simple biopsy is now this work of art and science. It's a vibrant landscape of blues, purples, and pinks, all on a tiny glass slide. And every color, every shape, and every pattern is a clue, just waiting to be interpreted by a trained eye. So what is the pathologist actually looking for in this cellular landscape? Well, they're basically detectives searching for any deviation from the norm. Are the cells too big or too small? Are the nuclei dark and irregular? Is the whole structure of the tissue disrupted? Are there inflammatory cells where they just shouldn't be? They're piecing all these visual clues together to understand the story the cells are telling, whether that story is about inflammation, infection, or even cancer. And that really is the magic of histopathology. It's this incredible journey that transforms a piece of biological evidence into a readable diagnostic map. It's the process that bridges the gap between a patient's body and a doctor's understanding, finally allowing us to see the story that's written in the language of our own cells.